So today we're going to pick up where we left off with this chapter, and so we're picking up with incomplete dominance. So when a heterozygote is going to have a phenotype that's intermediate between the two homozygotes, the example is going to be kind of a mixture. So not one's going to be dominant over the other, though you do have like a more dominant one or a capital letter and a lowercase letter. When you put those together, it's going to mix. So for examples, when you get these pink snapdragons that result from a cross between a white snapdragon and a red snapdragon. Or you can have a wavy hair texture as a result between a cross of a homozygous individual with curly hair and a homozygous recessive individual with straight hair. Codominance is going to result when you have a heterozygote and it displays characteristics of both alleles. And so it's not really a mixture, you see both. So examples of this could include feather color in chickens. When a white chicken or hen is going to be crossed with a black rooster, the heterozygote is going to exhibit both white and black feathers. Or you could have blood typing. So when you cross, let's say, a type A with a type B, you end up with an individual who could be AB. In that case, is going to be referred to as multiple allelism. And so with a pattern of inheritance in which a single gene has more than two alleles for a gene within a population. So example, you have your ABO system for blood groups, and that's going to be determined by 3 alleles. And you always use a capital I with the A and the B as subscripts on top, and then a lowercase I is going to code for type O blood. And so these are going to be the antigens that give rise to 4 different blood types. You could have A, B, AB, or type O blood. And so looking here, the allele or the genotype would be what you see up top in the alleles, and then coming down for the red blood cell antigen. If you have type A blood, you have those A antigens that are going to be in the cell membrane that are on the outside of that blood cell. If you have type B, you're going to have the antigens you see there that look like little diamonds around the outside of the cell membrane of that B blood cell. If you have AB, you're going to see both of those proteins that are on the outside, or antigens. And if you have type O, there's not going to be any antigens, kind of like a clean slate. Now with these, you can't mix these because if you have type A antigens, then you're going to have type B antibodies. So it will go in and it causes that blood to stick together and coagulate. If you have type B blood, then you're going to have type A antigens. And so they're going to come in, or antibodies, not antigens, sorry, antibodies. They're going to come in and they're going to cause it to coagulate or clump together. If you have type AB, then you have those AB antigens on the outside of that blood cell, so you'll have no antibodies that are going to come in and that are going to cause it to coagulate and clump together, so it can have blood from A, B, or type O. Now, if you have type O blood, you're going to have antibodies for type A and for type B, and so if it gets either of those types of blood transferred in, it's going to cause coagulation or clumping together. Some traits are going to be controlled with additive or multiple genes. So this is called a polygenic trait. That's a single trait that's influenced by the additive effect of multiple genes. So example with this would be height or skin color. And so you can see how you have multiple genes for this, and how when you put these multiple genes together, it could give you different skin colors. Pleiotropy is going to be a single gene that influences multiple unrelated phenotypic traits. An example of this would be like sickle cell disease. That's going to be a fatal condition in which defective red blood cells are produced with abnormal hemoglobin molecules and are ineffective at carrying oxygen within the body. This is done through one amino acid or one base pair mutation. Next, we have what's called sex-linked traits. These traits are going to be coded by a gene on a sex chromosome. So the X is where it's going to be found. So the first observed during Thomas Hunt Morgan's 1910 Drosophila experiment when he tested and crossed fruit flies, or gnats. So with sex-linked traits, the chromosomes carry different types of genes associated with both male and female reproductive processes. The X chromosome is going to be much larger than the Y chromosome, and it carries over 1,000 genes. More common in men than women because men only get one X chromosome, and the alleles on that chromosome are going to be expressed. So the X chromosome comes from the egg that is fertilized by a Y sperm. Now for females, they have both an X from mom and an X from dad, so they're going to have two sets of these chromosomes. So if one set tends to have, let's say, the colorblind trait, if they got a good set on the other chromosome from the other parent, then it will not express itself. They will simply be a carrier of that trait. So color blindness will occur when the color vision gene has an abnormal allele that cannot produce the light-sensitive proteins that are responsible for seeing red and green wavelengths. Hemophilia is going to be another trait. That's a blood clotting disorder that occurs when the body is unable to produce the necessary blood clotting proteins that are responsible for clotting the blood after an injury. So hemophilia is going to have been found in many prominent royal European families, and its occurrence is going to be traced back to the British royal family Queen Victoria. Usually, it does skip a generation, with a daughter of an affected male being a carrier and for that recessive trait, which she may pass to her son. This can arise when a specific group of dominant or recessive alleles are inherited, or when gametes contain an abnormal number of chromosomes due to errors in meiotic division. Nonspecific in nature, the occurrence on either autosomal or sex chromosome is going to be where we find these different genetic disorders. So let's talk about some of these in a minute. Pedigrees are going to be a useful tool that can trace the inheritance of a trait or filial occurrence of a genetic disorder over many generations. It can be used to determine or rule out a pattern of inheritance and predict the chances of an offspring having a particular genotype or phenotype, determine whether a trait's going to be carried on a sex chromosome or whether it's carried as a dominant or recessive on autosomes, for example, or it can also determine who may be carriers of a particular trait. So this is going to be constructed by obtaining a familial information over several generations. The information is going to be converted into a variety of shapes and lines. Males are going to be designated by squares and females by circles. If it's shaded in, the circle or the square, that means that individual will have the trait or genetic disorder of interest. Circles or squares that are going to be half shaded are going to represent carriers of that genetic trait or disorder. Horizontal lines between the squares and circles are going to be unions, so they're often referred to as marriage lines, even though it doesn't indicate that they were married. Vertical line is going to identify the offspring that's produced by that union, so the children. And additional horizontal lines are going to be added for each additional offspring. So looking here at this pedigree, you can see that we had Edward, Duke of Kent, and Victoria, Princess of Savoy. And so with this, you see how they're married. So you see that marriage line, or the union line is what they call it now, between the two. And you see that they had one child that's coming off of that, and that is going to be a female. Male is going to be square, remember. Female is going to be circles. So looking at this, we see Queen Victoria of England is going to be a carrier of the trait. She's married or has a union line, and you see all the different children she produces. So first, let's go down and look at all of the children she's producing. First one off, she has Leopold, Duke of Albany. That is a male. He is affected. So he does carry the hemophilia trait. Coming over, next, we have a female daughter, Alice. Alice is going to be a carrier of the trait, but does not exhibit the trait. Next, she has two more males that don't have that trait. Then she's going to have two more female daughters. These are going to carry the trait but not exhibit it. Next, you see we have King Edward. He doesn't carry that trait. Next, you see another female, and then a female that's going to be a carrier. And so altogether, 2, 3, 4, 5, 6, 7, 8, 9. She does have 9 children there. And so you see their unions, and you see how their children come off as either carriers, affected by that trait, or non-carriers. Autosomal recessive genetic disorders are going to occur when an individual inherits 2 recessive alleles for a particular disorder. An example of this could be Tay-Sachs. So the individual is going to inherit 2 recessive alleles that result in a defective hexa gene that's located on chromosome 15. This affects 1 in 3,600 individuals of the Jewish population who are going to be Central and Eastern European descent. Individuals are unable to produce the enzyme hexa, and that's going to cause lipids to accumulate in the lysosome of nerve cells. That buries the nerve cells in fats, so lipids. The characteristics are going to include impaired nerve signaling in the brain, neurological defects such as loss of sight, loss of hearing, and loss of motor function, could lead to paralysis, early death, usually by the age of 3 or 4. Cystic fibrosis is going to be another one. Individuals are going to inherit 2 recessive alleles that result in a defective CFTR gene, and that's located on chromosome 7. This affects 1 in 2,500 individuals of European descent, with 4% being carriers of this disorder. So a defective CFTR gene is going to produce incorrectly folded chloride ions, and the channel doesn't allow that chloride to exit the cell, and so that's going to result in an increased concentration of chloride and water inside of the cell. Characteristics here are going to include thick, sticky mucus that's going to be found in the lungs and the pancreas, impairs gas exchange in the lungs, as well as digestion problems and absorption in the small intestine. With treatment, individuals can survive into their 40s. Here, looking at this one, it shows you how that mucus is going to start to build up. When that mucus builds up in the lungs, it can actually trap bacteria and cause lots of bacterial infections. Pneumonia is going to be common here. Sinusitis, that's going to be an infection of the sinuses. That's going to cause inflammation, swelling, and lots of mucus and bacteria there. With their skin, the sweat glands are going to produce very salty sweat. The liver is going to end up with blocked bile ducts, which could cause a lot of pain. With the pancreas, you're going to have blocked pancreatic ducts. So it's not going to be able to put the pancreatic enzymes into the small intestine that help to finish digesting some of the larger particles and be able to absorb them. Inside the intestines, they can't fully absorb nutrients because of this, and reproductive organs, male and female complications can occur. Sickle cell disease is going to be where an individual inherits 2 recessive alleles that result in a defective hemoglobin B or beta gene located on chromosome 11. This affects 1 in 400 Black Americans. A defective hemoglobin beta gene is going to produce an abnormal hemoglobin molecule, which results in a sickle-shaped red blood cell. That's going to be fragile, and these die shortly after breaking apart as they pass through the blood vessels. Characteristics are going to include anemia, blood vessel blockages, joint pain, poor circulation, organ damage, physical weakness, and stroke. Individuals with sickle cell disease are going to be resistant to malaria, though. So the important thing about this, if you are a carrier of sickle cell, you don't exhibit the trait, you do have some sickle cells, but you do have some normal red blood cells, so they can carry oxygen, but it makes you resistant to malaria. So if you're bitten by a mosquito that's carrying malaria, you will not catch it. So this is why it's seen more in areas that are close to the equator where we have a lot of humidity and a lot of mosquitoes. Treatment options are going to include blood transfusions, bone marrow transplants, and CRISPR technology is working on this at the moment. That's gene editing. Autosomal recessive genetic disorders like sickle cell here are going to be shown. It's where one base pair is going to change, and so the amino acid changes. When that amino acid changes, it no longer produces the proper hemoglobin, and then you see those sickle-shaped cells in the slide here below. Autosomal dominant genetic disorders are going to occur when an individual inherits at least one dominant allele for a particular disorder. So some examples of this would be like neurofibromatosis. And so individuals here are going to inherit at least one dominant allele, resulting in a defective NF1 gene that's located on chromosome 17. It affects 1 in 3,500 newborns. So a defective NF1 gene is unable to produce neurofibromin, and that's a protein that regulates cell growth. Characteristics are going to include the formation of tumors. So they grow without stopping and checking themselves, they're going to create these tumors, and then those cells don't actually do what they're supposed to do. So the tumor is going to grow along the brain and spinal cord and cafe au lait spots. And in most severe cases, high blood pressure, scoliosis, and hearing and sight loss will occur. Achondroplasia is going to be where an individual inherits at least one dominant allele that results in a defective FGFR3 gene that's located on chromosome 4. It affects 1 in 40,000 newborns. This defective gene produces an overactive fibroblast growth factor for receptor 3 proteins, and that's going to interfere with the ossification of cartilage in the bone. Individuals born inheriting both dominant alleles will die prior to or shortly after they are born due to respiratory failure. That is called a lethal gene. Or if you've ever watched any of the Little People Big World TV shows, they talk about the double dominant, and they are always worried about that. That's what this is. So if you get both of the dominant traits, usually it leads to the body terminating the pregnancy on its own, or those babies die shortly after birth. So individuals, like we said, born are going to go into respiratory failure. And they also, with this gene, if you get one of these dominant traits and one recessive, it's going to include short stature, which we refer to as dwarfism, short limbs, a walking sway, macroencephaly in the prominent forehead, short fingers, ear infections, and sleep apnea. Huntington's disease, that's going to be where an individual inherits at least one dominant allele that results in a defective HTT gene that's located on chromosome 4. This will affect 1 in 10,000 individuals of European descent. So this defective gene is going to produce a longer than normal Huntington protein, and that consists of more than 40 trinucleotide repeats of CAG, which can build up in the brain. Function of the Huntington protein is going to be relatively unknown, but may be associated with normal brain development and cellular signaling and transport. Characteristics include neurological disorders that form later in life. They're going to start around the age of 35 to 40 years of age, and personality is going to change. They're going to start having poor coordination, muscle spasms, and imminent death within 15 to 20 years after the onset. So looking here, you can see that CAG in that section that's normally going to be there in the healthy gene. But if somebody has Huntington's disease, you see how you have 37 to 80 repeats of the CAG. And remember, you change the protein, you change the function. And so that's what's happening. Non-disjunction is going to result in gametes having an abnormal number of chromosomes due to unequal distribution during meiosis. So what happens is, instead of one of these chromosomes being pulled to either one side or the other, it could happen during meiosis 1 or meiosis 2. Then it's going to pull both of those chromosomes. And so one of these cells is going to end up, gametes, is going to end up with no genes in it, or no chromosome for that one, and one will end up with double. Now, depending on which one becomes part of the fertilization process, depends on if it's missing or if it's doubled up. Non-disjunction happens in autosomal genetic disorders, where it can occur in individuals that inherit an autosomal chromosome. So this is chromosomes 1 through 22. Now, it can happen in autosomal or sex chromosomes. We're going to talk about the autosomal first. So trisomy 13 is called Pitot syndrome. This individual is going to have an additional copy of chromosome 13, and it affects 1 in 1,600 or 16,000 newborns. Most individuals die shortly after birth, with only about 5 to 10% that live for more than a year. Characteristics include heart defects, nervous system abnormalities, a cleft lip, cleft palate, severe intellectual disabilities, small adrenal glands, and undeveloped or underdeveloped face. Next one, we're going to have Down syndrome. That's called trisomy 21. Individuals are going to have additional copies of chromosome 21, and this affects 1 in about 800 newborns. This is going to be the most observed chromosomal disorder in humans. About 50% die within the first year. Characteristics are going to include distinct facial features such as a flat face, slanted eyes, small mouth with a large tongue. They come with learning disabilities, a shorter stature, weak muscle tone, and increased risk for leukemia and Alzheimer's. With this, we see the non-disjunction and autosomal genetic disorders with trisomy 21 here. And so 21 tells you the chromosome, and you see that there are 3 there, so that's your trisomy. And this is looking, remember, this is called a karyotype that you're looking at. Non-disjunction on the sex chromosomes is going to where you have 2 copies normally of a sex chromosome, but here you either are missing one or you get a 3rd copy. So with Turner syndrome, females are only going to have 1 X chromosome, and they're not going to have that 2nd sex chromosome. This affects 1 in about 2,500 females. Only disorder in which a person can survive without 1 pair of chromosomes. So this one is survivable. Characteristics include varying degrees of intelligence, shorter height, webbed skin around the neck and shoulders, hormone deficiencies, underdeveloped sex characteristics, sterility, which means they can't produce offspring, and cardiovascular issues. Hormone replacement therapy is going to be an option for these people and allowing sex characteristics to develop with them. Next, we have trisomy X. This is where they're going to have 3 X chromosomes. So either the egg had 2 X's and then they had an X in the sperm, or the egg had 1 X and there's going to be 2 X's in the sperm. This is going to affect 1 in 1,000 females. Characteristics include being taller than normal, thin, varying degrees of intelligence, normal sexual development despite their irregular menstrual cycles, and most do conceive offspring. Sometimes you might see this called super female or mega female. Next, we have Klinefelter syndrome. That's where males have 2 X chromosomes and 1 Y chromosome, and they are denoted as XXYs. This affects 1 in about 650 males and the most common genetic abnormality in humans. Characteristics include being taller than average, some learning disabilities, some hormone deficiencies, underdeveloped testes, sterility, and development of female features. Also, you might have high blood pressure, type 2 diabetes, and rheumatoid arthritis. Now, with the female features, you might start to see more rounded breast tissue or chest, more rounded hips, a rounded face, just kind of more softer look to these males. Hormone replacement therapy is going to be an option, but this only stimulates muscle strength, libido, and reduction of that female development, and has no effect on the testicular development or the sterility. Jacob syndromes, where males have 1 X chromosome and 2 Y chromosomes, and is denoted as XYY. This affects 1 in 1,000 males. Characteristics include being tall, some 6 feet or taller, slightly lower intelligence, high levels of testosterone, and development of male secondary sex characteristics is going to be normal, and most are able to conceive offspring. This one's going to be referred to as the super male, meta male, or even has been referred to as the Johnny Bravo syndrome. That's where you have the super jock or the person who's super sporty, super tall, super athletic, and not quite as smart. And that's going to end us for this unit. We'll see you back for the next unit.